Ain Shams University, Egypt in 2010. Then he moved to Germany to get a Master of Science in the field of neuroscience from the University of Göttingen in 2013. In 2020, he got his PhD from University of Georgia, USA. After that, he continued his postdoctoral research for almost 2 years. He published in high-profile international journals such as Nature Scientific Reports, International Journal of Nuclear Sciences, PLOS ONE, Transitional Stroke Research, and so many. You can, of course, check his Google Scholar in our website. I am sure that we will be enriched today by the seminar. Once again, I welcome all of you, and let's start our seminar with Dr. Mohammed. Dr. Mohammed, thank you for accepting the invitation, and I will kindly ask you to start your presentation.
Thank you very much, Dr. Salam. Good morning or good evening, everyone, depending on where you are. My name is Mohammed Sayed, and like Dr. Salam said, I am an assistant professor of pharmacology in the department of the Bas College of Osteopathic Medicine in Lincoln Memorial University. And today I'm going to speak to you about this paper from 2017, which discusses the use of induced pluripotent stem cell-derived neural stem cell therapy in the treatment of experimental stroke. Some of you might ask, why did you specifically choose this paper from 2017? And the reason for this is because during my postdoc years, I worked with this same group. However, this same group stopped publishing papers after this one because they became a company. So you know, when you publish something in the public domain, you stop having the intellectual property of this of what you published. So because this group turned into a company, this company is called Aruna Bio right now. Whatever they reach and all the results of the research, it is under copyright and under intellectual property protection, and they do not publish it anymore. So this was one of the last paper they published, the last papers they published, and I decided to share this paper with you specifically because it does not, it's maybe more relevant to the, let's say, molecular biology people among you, rather than the pharmacology or something like that. This is a small introduction about myself. Like Dr. Salam said, I got my bachelor from Ain Shams University in Egypt in 2010, my master's from the University of Göttingen in Germany in 2013, and my PhD from the University of Georgia in 2020. And I worked as a postdoctoral fellow at Aruna Bio from 2020 to 2022. And then last year, I started working as an assistant professor in Lincoln Memorial University. Yes?
I'm sorry, can you share your presentation?
Yes. The presentation, I will submit it to Dr. Ahmed Salam, and he will send it to the group.
No, not here.
You cannot see it in front of you?
I can see.
Dr. Salam, can you see my presentation?
Yes, I can see. I can see your presentation, but
so what's the problem?
okay, so I think no. Yeah, go ahead. I have seen your slides. No problems.
Can you see my mouse pointer right now?
No, it's freezed.
Okay.
I think, as I told you, that it may be better to share your screen because you know some technical problems may happen for sharing slides.
Okay, let's share our screen. Okay, is it better now?
Yes, yeah. I think, yes, it's better.
Can you see my laser pointer?
Yes, Dr. Mohammed.
Yes, it's clear.
Okay, so this is my just an introduction about myself, and this is the link for my Google Scholar. And like I said, I will also give the slides to Dr. Ahmed Salam, and he will share it with you all after the presentation. Now, let's speak about stroke a little bit, because what we are trying to treat here is stroke. Stroke, which is the second leading cause of death worldwide. Okay. In the United States, it used to be the third leading cause of death, but with time, things became better, and you can see the graph going down from the mortality per 100,000 residents until it now became the fifth leading cause of death. However, because of so many people who have stroke and they survive stroke without dying, most of these people become disabled. So this disability can be motor disability, for example, people having paralysis or paralysis or something like this, and it can also be is there are no approved treatments for stroke other than a specific treatment called RTPA, which is tissue plasminogen activator. This is a drug that is given to people who are currently suffering stroke to dissolve the blood clot that blocks the blood vessels in the brain to resolve the stroke. The problem with this treatment is you can only give it in the first 3 to 4 hours, maybe up to 12 hours after stroke. But the problem is most of the people who suffer stroke, by the time they go to the hospital, this treatment window, the opportunity passes and they cannot receive this treatment anymore. So we need to find new treatments that can help patients who actually suffered from the stroke, but they did not receive the RTPA. Now, stroke can be one of two things, either ischemic stroke, in which a blood clot or something else
Dr. Mohammed,
Hello?
ايوه مش بيفصل عندك.
Okay,
خلي بالك.
let's try this again.
Yeah.
Okay. Can you see me right now?
Yes.
Can you see the slides?
The slides and your mouse.
Okay, so we were discussing stroke. So I was saying that the stroke can be one of two kinds. Either an ischemic stroke, in which there is something that blocks the blood vessels of the brain, whether this blockage can be a blood clot or it can be also an embolus. An embolus means like a piece of fat or a piece of anything that actually goes and blocks the blood vessel. And in this case, not enough blood reaches the brain, causing the death of the brain cells. And we call this an ischemic stroke. And it is 87% of the kinds of stroke. We also have something called hemorrhagic stroke, in which a blood vessel inside the brain will break and start leaking the blood outside the brain. When this happens, also there is a part of the brain that does not receive the perfusion, the amount of blood necessary to survive, but also the brain that is getting leaked out in the skull or outside the tissue puts pressure on the other parts of the brain, causing more and more damage. And this is why this hemorrhagic stroke, even though it is only 13% of the people who suffer from it, but it carries the majority of risk for death after stroke. So most people who die, who get hemorrhagic stroke, actually cannot survive afterwards. Okay, please, I know that some of the people who are attending here are not from the medical field. So if you find that any of what I'm saying is very difficult to understand, please stop and ask me, and I'll try to answer it in an easy way. Now, what happens after ischemic stroke? We'll focus on ischemic stroke here. So what happens is in the first few minutes after stroke, you can see inside the brain, there is a core of the stroke. This core of the stroke is the cells that die already. And they die because they are not getting enough oxygen, they are not getting enough nutrients from the blood. And the brain is a very metabolically hungry system. It consumes a lot of energy. And if you do not supply the brain with the energy it requires, it dies very quickly. And when it dies due to mitochondrial dysfunction, which is a problem in the respiration of the cells in the brain. This causes oxidative stress, and this oxidative stress kills some of the brain cells. When these brain cells die, they release the neurotransmitters inside them, causing the other cells around them to get too excited. And when they get this too excited, we call this excitotoxicity. These cells that became too excited will be drained from the metabolites also very fast. And since they are not getting enough oxygen and enough metabolites, they will also start dying. So it becomes a chain reaction. And during this chain reaction, what happens is that this core, which starts in the brain as a very small area, starts increasing in size with over hours and even over weeks, until what happens is, instead of a small injury like this, you get a huge injury in the whole brain. What we are trying to do is trying to stop this transition from having a small core. This core is already dead, so you cannot save it anymore. But we do not want this penumbra, which is the area that can become dead, to die. So, and this is called neuroprotection. Neuroprotection is that we are trying to prevent the neurons in this penumbra from actually dying through the oxidative stress, excitotoxicity, and post-ischemic inflammation that happens. Okay. Now let's speak about stem cells. I know most of you are molecular biology people, so you know about stem cells probably more than what I know. But just to give an introduction for those who are not very familiar, stem cells are those kinds of cells in the body that can differentiate to produce different kinds of other differentiated cells. So the biggest stem cell, the most important stem cell, happens when a person is an embryo. When the zygote is formed by inside the mother after fertilization, it is a totipotent stem cell. Totipotent means it has the potential to give the total body of the human being or of the animal. Okay. After a while, these totipotent stem cells, like here the blastomere, it forms into something called embryonic stem cells. The embryonic stem cells are usually pluripotent. Pluripotent means that they can differentiate to give many different kinds of differentiated cells, but without the ability to differentiate to become the whole organism. And then these embryonic stem cells can differentiate into mesenchymal stem cells, hematopoietic stem cells, and neural stem cells. And each one of these three is a multipotent. Multipotent means it can differentiate to give only a few kinds of differentiated cells. So, for example, mesenchymal stem cells can give, through osteogenesis, can give bone, cartilage, muscle, and for example, fat and skin, and so on. Hematopoietic stem cells can differentiate to give white blood cells, red blood cells, platelets, and other kinds of blood components. Neural stem cells can differentiate to give either neurons or oligodendrocytes or astrocytes. Okay, so the problem with the brain is, once the brain reaches the final differentiating state, where we have neurons, oligodendrocytes, and astrocytes, all these cells do not divide anymore. So whenever you lose a cell out of these, like you lose a neuron, you lose an oligodendrocyte, you lose an astrocyte, you are not getting any replacement. This is what makes brain injury very dangerous, and this is also what makes stroke very dangerous, because it's a kind of brain injury. What we are trying to do here is using a specific technique called induced pluripotent stem cell, which was discovered in Japan in 2006 by Takahashi, and it was a very let's say, revolutionary technique. We can take some of the normal cells, fibroblasts from the body, and treat them with these factors. And once you put these factors on these stem cells, it gets reprogrammed to produce pluripotent stem cells. So pluripotent stem cells are the stem cells, the same ones that you can find here. They are very similar to embryonic stem cells. Of course, there is a very big problem with using embryonic stem cells in research, because embryonic stem cells, you have to take them from an embryo, and if you take them from an embryo, there is all these ethical dilemmas that are related to it. But by using this process, redifferentiating fibroblasts into pluripotent stem cells, what happens is that you get something that is very similar to embryonic stem cells. Now, these embryonic stem cells, using other factors in the lab, we can turn them, we can differentiate them into things like cardiomyocytes, adipocytes, neurons, beta cells of the pancreas, and so on, all the different kinds of cells that we want to do. So this technique is called induced pluripotent stem cells, and it revolutionized medical research in the last 15 years. Now, we are going to focus right now here about a specific kind of these cells, which are called induced neural stem cells. So after we get the pluripotent stem cells, we give this stem cell some soluble factors, and these factors cause it to differentiate into a neural stem cell. Now, the neural stem cell itself can differentiate further into either becoming a neural progenitor, which means the progenitor of the neural cells producing neurons. Neurons are the main cells of our nervous system. Or they can also differentiate into something called glia. Glia is the support cells in the nervous system. So our nervous systems as humans contains about 1 billion neurons, but our central nervous system, and it contains about 10 billion glia. So glia can be oligodendrocytes or astrocytes. I will not go into details, but I just want you to know that these cells are also very, very important to the health of the nervous system. And also these cells, if they are lost, we cannot replace them anymore. Also, these neural stem cells can themselves produce soluble factors. Not only the benefit of the stem cell is not only producing, turning itself into a specialized cell, but it can also produce soluble factors that can help maintain the health and the integrity of the brain tissue. And usually this is being produced in the form of exosomes. Exosomes are extracellular vesicles. These vesicles contain a cargo inside. This cargo can have proteins, nucleic acids, and many other metabolites. And it is covered with a double membrane, like the plasma membrane, and including some transmembrane proteins, as well as receptors and so on. This package that can be produced from the stem cells can contain bioactive molecules that can help the brain, can help the cells communicate with each other, as well as helping the brain regulate its function and repair the damage that happens to the brain. Okay, put this here so you can see better. Okay. Now, let's look at the experimental design of our paper that we are discussing today. So in our paper, we have a pig model, and this pig, we induce a stroke in the pig. We induce this stroke in the pig by opening the brain and putting something that would block the blood from reaching specific parts of the brain, causing a stroke. And then, similar to the normal stroke that usually dissolves, we then remove this plug that we put in the brain to simulate what happens when a human gets a stroke. Okay. We generate the induced neural stem cells in the lab, and then we make sure that these stem cells are actually neural stem cells. And on day 5 after stroke, we transplant these stem cells into the damaged areas in the brain of the pig to see if these stem cells will actually help the pig recover from the damage of the stroke. We confirm this by doing an MRI at day 1, week 1, week 2, and week 12 after stroke to see the short-term effects of the implantation, the transplantation of the neural stem cells, as well as the long-term effects at 12 weeks after the transplantation. Later on, we sacrifice the animals and do the histological analysis on these pigs. Now, in our paper here, I will not go, the paper has 9 figures. I will not go over all the 9 figures, but I will take the important things that you can see from this paper. Now, the first thing that happens here is that we have to make sure that the stem cells that we are actually measuring are actually neural stem cells. So from an induced pluripotent stem cells, we want to make sure that the differentiation occurred correctly, and these pluripotent stem cells can turn into neural stem cells. And for this, they used flow cytometry after treating the cells with the specific factors. And with flow cytometry, they are measuring the levels of SOX1 and nestin. Nestin and SOX1 are both markers for neural stem cells. And you can see here from this results, this is the control, and this is our test groups. We can see here like 94.8% of induced neural stem cells co-expressed both the SOX1 and the nestin, which makes us 100% sure that these cells are neural stem cells. Okay, and we can see this not only through flow cytometry but also through microscopy. SOX1 here is green, and DAPI, which is the marker for the nuclei, is blue. And you can see here that these cells express SOX1 as well as nestin. So with the co-localization, you can see that all these cells have SOX1 and nestin. You can see from this figure, you can notice that SOX1 is nuclear, so it coincides with the nucleus, while nestin is cytoplasmic, so it goes into the cytoplasm. Okay. Now, after making sure that these cells differentiated into induced neural stem cells, we want to make sure that they take the next step into the differentiation, and they can differentiate into the different either neural or glial differentiated cells. So first of all, we measured the amount of TUG1. TUG1 is a marker for neurons, and we can see that a subset of these cells actually express TUG1. Also, MAP2 is a marker for neurons, and you can see that some subset of these cells, like this cells here, for example, has the yellow color, which means it produces both TUG1 and MAP2. So we are sure that these stem cells can produce neurons. We are also looking at GFAP. GFAP is a marker for astrocytes. Astrocytes is a type of glia, one of the support cells of the nervous system. And we can see that the subset of these cells also can produce astrocytes. And OLIG2 is the marker for oligodendrocytes. Oligodendrocytes is the marker for the OLIG2 is the marker for oligodendrocytes. Oligodendrocytes are the cells responsible for producing the myelin sheath in the brain. The myelin sheath is insulation material. If you think about the neurons as being cables, and these cables carry electricity from one part of the brain to the other, or from the brain to the rest of the body, these cables require insulation, so the cable must be insulated, and for this insulation to happen, you need oligodendrocytes. So our stem cells can produce neurons, and they can produce GFAP positive cells, which are astrocytes, as well as OLIG2 positive cells, which are oligodendrocytes. So we are sure, we now validated the model, and we are sure that our cells here are actually functional neural stem cells. Now let's look at the stroke. We want to make sure also that our stroke is successful. So we have 8 pigs. 4 are going to be, all of them are going to be stroked. 4 of them are going to receive the neural stem cell transplantation, and the other 4 are not going to receive it. And you can see here in the panel A that this is an MRI of the pig brain, and you can see here this white area. This white area is the area that got affected by the stroke. So it's either dead or dying. Okay, and you can see here. And you can see in the second kind of MRI image, the swelling inside the brain. So here in this second kind of MRI image, water appears dark. So you can see that this area, which is a little bit bigger than the area that you saw in panel A, contains a lot of water. So when it contains a lot of water, it means there is edema. Edema means a swelling inside the brain. So by using this MRI method, we validated that actually the stroke was successful and the pigs got stroked. Okay. Now, afterwards, we are going to look at the difference between the treated and untreated. You can see here in the panel C. So the panel C here shows us first of all, before going to treated versus untreated, we want to see how the stroke lesion resolves over the course of the 12 weeks. So here in panel C, you can see in the untreated animals, the brain swelling, the initial brain swelling 1 day after stroke. And here you can see 4 weeks after stroke. And here you can see 12 weeks after stroke. So 1 day, 1 week, 4 weeks, and 12 weeks after stroke. And you can see how first, this is a huge swelling. This swelling goes away after 1 week. So this is the initial part of the stroke. The swelling goes away. But then afterwards, this brain tissue dies and it starts becoming liquefied. We said that the dark area here is liquid. So it starts becoming liquefied until it disappears completely after 12 weeks. So those animals who have a stroke, after 12 weeks, basically, if you open the brain of the animal, you will see a gap, like a big vesicle. This vesicle is empty. It does not contain any brain tissue because all of the brain tissue inside it died. Okay, this is important because by judging the size of this lesion here at the different time points, we can know whether our treatment actually worked or it did not work. Okay. Now, this is the comparison between the volumetric measurements for the size of the stroked hemisphere. You know, the brain has 2 halves. We call each half of these a hemisphere. So we compare the size of the stroked hemisphere compared to the contralateral hemisphere. And what we are going to see here is that those in just 24 hours after stroke, the 2 hemispheres are actually so you can see here on the y-axis, we are comparing. This is a % increase compared to the contralateral hemisphere, which means like for the non-treated at 24 hours, you can see here that the brain, the part that is actually stroked, became bigger. So how can the part that gets a stroke becomes bigger? It becomes bigger because of the swelling, of the edema. There's a lot of water that comes from, there is an inflammation, a lot of water that comes from the part of the body, and it pushes the brain. So you can see here that what happens is the brain becomes bigger at 24 hours. Later on, 1 week later, this edema disappears, all this water goes away, and the brain, the half that got stroked, becomes equal in size to the half that did not get the stroke. Later on, what happens is shrinkage, and this is what we discussed here, that we lose the brain tissue. Okay, so what happens is that the part that gets the stroke starts shrinking, and when it starts shrinking, it means that we have more and more tissue dying. You can see here that the non-treated compared to the treated, the shrinkage in the treated is a little bit less than the non-treated. Okay, so these are not significant results between them, but it is a trend that you can see that the treatment actually succeeded in reducing the shrinkage of the brain to a very small extent. What was more significant for us right now is the change in white matter. So these figures here show you the white matter, a way we measure the amount of white matter in the brain. It's called DTI. And the amount of white matter, that, like I explained, the white matter is the insulation of the brain. So when you think of the brain as if they are wires, the neurons in your brain are wires that are going everywhere. These wires must be insulated from each other. So because if they touch each other, they will cause problems. So the more insulated these wires are, the better the signal transduction inside the brain. So we can see what happens is that after stroke, the hemisphere that got stroked actually loses some of this insulation. So you can see that the amount of insulation is either 20% to 40% less. And with treatment, the part that got the treatment actually returned significantly towards the base value compared to the part that is untreated. So our treatment here, the transplantation of these stem cells, actually successfully produced remyelination. Remyelination means the production again of the myelin sheath, of the white matter, the insulation of the brain cells. This is another thing that we measure the stroke with it, which is the blood perfusion. We said that when stroke happens, the blood stops going to the brain. In this image, you can see the blood, the parts that are perfused by the blood, as being the light color. So either green, red, or yellow. So if you make a line in the middle, you can see that to the right, which is the part that is unstroked, has much more blood perfusion compared to the left in the non-treated. Here in the treated, we get a little bit more perfusion. Of course, it's not back to the baseline that you see here in the non-stroked, but it is much better than what we get with the non-treated. So here we can see from this figure that the neural stem cells actually successfully produced improved blood perfusion in the stroked hemisphere. And when we quantify this, you can see here, this is the change in cerebral blood volume. You can see here that with time, the treated animals started having an increase in their blood volume, and this increase actually remained stable until 12 weeks after stroke, while the non-treated animal, the levels of the blood in the stroked hemisphere was actually kind of the same. It was plateaued. We also wanted to look at the amount of the different kinds of cells, whether they are neurons or oligodendrocytes or astrocytes or microglia inside the part that has the damage. And you can see here, this is the normal amount of neurons. And for those animals that did not receive the treatment, the amount of neurons decreased significantly. And then when they receive compared to the animal that received the treatments, you do not get the same reduction in the amount of neurons, which means that the treatment actually successfully rescued some of the neurons from dying. On the other hand, when we look at IBA1, which is a marker of inflammation, inflammation means that the immune cells of the body start infiltrating the brain tissue. This is not a good thing, because when you have these immune cells going into the brain tissue, they start breaking the different tissues in the brain, killing the other neurons, putting pressure on the metabolism of the brain. So we actually want this to be low. And we can see here with the normal brain, it is 0%. But then once you get the stroke, the non-treated, it goes really up. And when the animals get treated with the neural stem cells, this goes down again. Not to the baseline, but significantly down compared to the non-treated, which tells us that the treatment that we did actually was successfully reducing the inflammation. Comparing GFAP, which is the amount of astrocytes, astrocytes are beneficial cells, but also they increase in size once they sense inflammation. So we can see here with astrocytes that this is the normal amount of astrocytes, and after stroke, whether we are treated or non-treated, you get the same increase in astrocytes. So our treatment here was not successful at reducing astrocytosis, which is the normal response of the body in the brain to inflammation. Now we want to look at these cells that we transplanted. Did they actually differentiate to produce neurons or astrocytes or oligodendrocytes, or did they remained as if they are stem cells? We want them, of course, to be differentiated. So here, HNA is the human nucleic antigen, which is a marker for human cells in general. Usually, because this is a pig brain, when you look at the pig brain, you shouldn't see any HNA at all. But because the stem cells that we transplanted into the brain of the pig was actually coming from humans, so we can see here some markers that say that these stem cells actually remained for 12 weeks inside the brain of the pig, and the brain of the pig did not get rid of them. Some of these cells are actually neurons. So the NEON is a marker for neurons. And of course, you'll see a lot of it in the brain of the pig because the pig already has neurons. But we are interested in the cells that can express both. So it has both the green and the red. And these cells that you can see here, like this one, like this one, like this one, it means that these cells, which originally comes from a human lineage, it is also a neuron. This is an evidence that our cells here successfully differentiated into neurons inside the brain of the pig. Similarly, we can see here OLIG2 is a marker for oligodendrocytes, and we can see here that there are some cells that are expressing both human nuclear antigen and OLIG2, which means that they successfully differentiated to produce oligodendrocytes, which are the insulation cells inside the brain of the pig. Finally, I would like to discuss some of these results. The significance of this study is that many experimental treatments for stroke succeed in rodent models, but fail when they translate to humans. I myself, during my PhD, worked on rats. Rats are rodents. We usually work with rodents because they are very easy to breed and very easy to keep and very cheap, and you can get a lot of them. But the problem is that there is a big difference between the brain of the mouse or the rat and the brain of the human. So sometimes we use some treatments in the rats and the mice, but when we translate it into the human, it does not work. This study is unique because it uses pigs. The brains of pigs are much more similar to the brain of humans compared to the brains of rats and mice. Which means that there is a bigger chance if we use this treatment on a pig, that it would actually succeed in the human when we use it in the human. This study provides evidence for histological improvement that remains for a long time. It's very important for us that whatever improvement that we see, it is not a short-time improvement, but that it remains for a long duration. This makes us sure that when we put it in the human, it will work on the long term, and it will cause the situation to improve. And also that this study showed a beneficial effect of neuroinflammation. Neuroinflammation is something very, very dangerous, because when it happens, these infiltrating immune cells that comes from the outside the brain and go inside the brain, they start damaging the brain. So this study actually produced reduced neuroinflammation, which is also a beneficial thing. However, there are two very big limitations of this study. The first thing is the small number of animals, only 4 treatment animals per group, which is extremely, extremely small compared to what we do in mice and rats. In mice and rats, you can get 10, 12, even 20 animals per group. The problem why here we have only 4 animals per group is because pigs are big, they are expensive. They are expensive to work on, they are expensive to maintain, they are expensive. So it is very, very difficult to have a bigger number of pigs, and that there were no behavioral testing after stroke. So in this study, they did not actually test the functional outcome of the stroke. You know, when someone gets a stroke, they can get paralysis, they can get problems in their function. So in this study, they did not measure any kind of functional outcome for the pigs. Thank you very much. This is the end of my presentation. I hope I did not exceed the allotted time. And this is just a reminder for yourself that when you suspect someone that has a stroke, you gotta act fast. And fast stands for when you see a problem in the face, you see one side of the face is drooping like this, Or you see a problem in the arms, you tell the patient to hold both your arms together, and you see that one arm is going actually lower than the other. Or you see a problem in the speech, they are trying to speak, but the speak becomes very difficult or unintelligible. You cannot understand what they are saying. You gotta call 911 or call the emergency, because these are very, very critical time to save the patient. You gotta get the patient to receive the treatment within the first 6 hours. Otherwise, the damage is going to be permanent and you cannot fix it anymore. Thank you very much for your listening.
Thank you, Dr. Mohammed, for this really informative seminar to us. And now we open the discussion. So if you have questions, we can unmute your mic, and please you raise your hand in the chat, or you can write your question in the chat room, and I will tell it to Dr. Mohammed. So any question from attendees? Okay, I may start, Dr. Mohammed.
Yes.
That in the first diagram that you presented about the differentiation of stem cells, and you said that we cannot use stem cells from human due to ethicals in research, human rights, something like that.
Yes.
What about if we can use it from monkeys?
Yeah, there is it's the same ethical issue. So what do you, if you want to get embryonic stem cells, how do you actually get these embryonic stem cells? You have to take the embryo while the embryo is actually being formed, and then you harvest this embryo and actually make it into cells that you use for cell culture. Now, does this embryo have rights as a human being, if we're speaking about human embryos, or not? So if you are going to get a sperm, and this sperm goes into the ova and it forms a zygote. This zygote is right now a human life. So you cannot take it and actually use it to do research. Okay. Even though for some countries, like for example, China or Korea, where they do not consider these as human life, it is very difficult now. You will have people to work as human farms. Like you have to get a woman, and this woman you get her pregnant, and after sometimes you take out all the embryos from her body to use for research. So it is actually something extremely bad. Can we do this with monkeys? We can, but the problem is monkeys are like humans. They take a long time to become pregnant, and their pregnancy lasts very long, and then they are very expensive to maintain and very expensive to work on. And the problem is, after you get the cells from the monkey, you are not sure that it's actually going to go into the human and cause no problems, right? Because sometimes you might have allergic reaction, for example, to the cells if they are coming from outside. So the best way to do this is to actually take a cell from a person and then implant this cell into the person himself. So I will take a somatic cell from myself and differentiate it into a stem cell. And after it becomes a stem cell, I can implant it again into my own body. This way, I'm 100% sure that no allergic reactions will happen, that no different problems will happen due to this implementation. Okay, if you implant something from a different species, it's called a xenotransplant, and xenotransplants are very dangerous when it comes to causing problems inside the body.
Okay, we have a question in the chat room. So can you see it, or shall I read it to you in the chat?
Okay, Islam Abu Bakr. These difficulties also apply to the other body tissues, not just the neural cells. Yes, there are some parts of the body that can regenerate. When the regeneration happens, it's easy. For example, I'll give you an example. If we are speaking about the liver can regenerate. If you take parts of the liver, the liver can produce again more liver tissue. there are some kinds of tissue in the body that cannot regenerate anymore. And one of these tissues, he gave an example here, like cartilage cells. Yes, cartilage cells, if you remove the cartilage from someone, from the joint in the body, it does not regenerate again. So this actual stem cell treatment is very, very promising for treating things like that. If we can put the stem cell from your own body into your joints and then watch as these cells differentiate to produce more cartilage, then this would be the perfect thing. So we are very optimistic, if we can say so, about the use of stem cells, not only in the brain, but in the heart, in the muscle, in the cartilage, in the kidney, in any kind of tissue that does not regenerate anymore, which is the majority of the body tissue, of course.
Okay, thanks. Any question? Okay, Dr. Mohammed, regarding to the treatment on animal, rats and pig,
Yes.
how similar is between human brain and the monkey brain?
Yes.
If you get an approval, and if you get big fund, and you think that it can work with experiment on monkey brain, or
Definitely. The monkey brains are much closer to humans compared to pig brains. But if I tell you, for example, that pig brains are extremely expensive to work on, imagine how expensive it is on monkey brains. If you remember, when we were living in Göttingen, there was a place called the German Primate Center. The German Primate Center in Göttingen was actually focused on doing these kinds of experiments on monkeys. The problem is, for a monkey to become an adult, like if you have a new monkey, you see, when I want to produce new rats or mice, it will take me about 2 months from the rat or mouse being born until they become an adult I can work on. Okay. Do you know how long would it take me to get a monkey as an adult?
no?
It is exactly like a human. You will have to wait 10 years in order for the monkey to be an adult. So the problem is, when you want to work with monkeys in this situation, you have to harvest monkeys that are already adults right now, right? You cannot actually, I'm going to start an experiment right now. Okay, so generate 10 monkeys for me, and I can wait 100 years for these monkeys to come. No, you have to get monkeys that are already adults. And where are you going to get them from? Are you going to get them from a zoo, or are you going to get them to hunt them from the forest? It's very difficult. And there is also another ethical issue, is that monkeys, because they are very close to human, also they can feel emotions very similar to humans. So in many parts of the world, you are not allowed to work on monkeys, because whenever you try to work on monkeys, you are causing them to suffer. While we humans assume that things like pigs are going to be less developed when it comes to the mind compared to monkeys, so they are not going to feel emotion or suffering as bad as monkeys. Also, the pig has a other very good benefit, is that the size of the brain of the pig is very similar to the size of the brain of the human. So, for example, monkeys can differ in size. If you take a very small monkey, like you see, we call it in Arabic snas, yeah, it has a very small brain. But the size of the pig of the brain is the brain of the pig is almost exactly the same size as the human. This is one thing. Also, the immune system of the pigs is very similar to the immune system of humans. So the pig solves a lot of problems. However, still the pig is still very expensive to work on. So the majority of the labs are not going to be working with pigs.
Okay. Yeah, I have also another question.
Yeah, go ahead.
You have examined 4 animals, and as you said in your discussion, this is like small experiment or small number of animal for a trial. Did you notice any like differences among the 4 animals in the response to the treatment, or do you think if this response may be due to the genetics of?
Yes, definitely. There is some. I will try to share again, because I want you to show a specific slide. Let me share the screen. let's go to any other, let's see, for example, this graph. You can see the graph now?
Yeah, I can see, yes.
So in this graph, you see this huge bar of variability. This bar is very huge. It means there is a huge intervariability between the animals from the same group, right? Normally, you don't want to see this because when you have this huge bar, it means that whenever you are trying to calculate the p-values, you will not get good p-values. Now, the more you add animals, the more you increase the n in size, you will see that the variability will go down, go down, go down until you see no variability at all. This is what I want to see. If I'm going to do an experiment, this kind of variability is what I want to see. The problem, of course, there is a huge because we are not using mice or rats where you can design them to be as genetic. The genetic component can be designed as you want. There is going to be variability, and the only way to go around this variability is to increase the number of animal used. And increasing the number of animal used increases how expensive the study is. So it's really complicated. However, I can tell you about the future directions of this because, of course, this was in 2017. Now we are in 2023, so we have moved for the last 6 years. So in the movement over the last 6 years, we decided that we are not going to be transplanting the stem cells themselves anymore. So what is going to happen right now is that we culture the stem cells in cell culture. And then these cell cells, like I said, they produce exosomes. And these exosomes, when they are produced in the liquid by the cells, and we can harvest these exosomes, and then we can inject them into the brain, into the blood stream for the test animals. And this have produced very, very successful results so far. And it solves a lot of the problems. So right now, the company, Aruna Bio, is focusing on producing these exosomes and using them to target different brain tissues. And actually, the IND, which is the approval from the FDA to start the experiments on humans, is expected to happen this year. So after this year, we'll start actually using these exosomes to treat humans directly. And once you treat humans directly, it will be much, much easier and cheaper than treating pigs or monkeys or anything, because you are not going to kill the human to see and open his brain. What you are trying to do after when you are treating the human is you see a human who has a stroke, you give them the treatment and you see whether they improve or not, which is going to be much simpler when it comes to cost. Of course, it's still going to be very expensive. So doing a proper clinical trial on humans is going to have to cost at least $1 billion. But if this FDA approval comes, then we are hopeful that a big company like Roche or Pfizer or anything will buy actually this product and will start using it on humans in clinical trials.
Hope it works.
I have a question
Yeah,
from Islam Abu Bakr. I guess the AI can shorten the period of experimental examination when we are needing a ton of parameters to process. Do the AI can be very optimistic to do in a measure stem in the stem cell success? Okay, I will answer your question. Yeah, Islam. AI for the problem with our AI is good for a proof of concept. Like AI can get you whether the concept is worth testing or not. But actually, when you want to approve a drug or when you want to approve a treatment to use, you still have to test it. And the biggest problem is not in the lack of concepts. We have so many concepts that have been published to work on other animals, to work on cells, to work in rats, to work in mice, to work on pigs, and so on. We have a lot of concepts. The only problem is who is going to pay the $1 billion bill to actually test these concepts on a human, right? Because the drug cannot be approved to be used in the market on humans unless the people actually, the FDA gives it an approval, and the FDA will not give it an approval until it gets tested on humans. So let's see, you are a pharmaceutical company, you are Pfizer, for example, and in front of you, you have 100 different promising candidates that you want to test. But in order to test each of these candidates, you'll have to pay $1 billion. So you are not going to pay $100 billion to test all of these, and only one would be successful, and maybe not all of them are going to be successful at all. So what you should be doing, what you are trying to do is you get the most promising one through animal experimentations, and this one you put your money on it and gamble on it and see whether it's going to be successful or not. So short answer, yes, the AI can help in the early process, which is the process of proof of concept. But once the concept is actually proven, the biggest hurdle against producing new treatments is actually the human experiments, which are very, very expensive.
Okay, thanks, Dr. Mohammed. So, do we have any questions?
Yes, I would like to thank you all for inviting me to give this lecture, and I hope it has been beneficial for you, even if you are not from the medical field. Maybe you got some idea about how to test something like stem cells in actual experiments. If you have any questions, please do not hesitate in contacting me through email, and I will send the slideshow to Dr. Ahmed Salam so that he would actually share it with you if this is possible, Dr. Salam.
Yeah, actually, all info about Dr. Mohammed is present in our website. So his link, his email. So please, if you would like to contact him, please go to our website and you will find all information as well. And I think the poster of Dr. Mohammed is has all info, and also you can visit our website. Okay, thank you, Dr. Mohammed, again for this nice presentation. And for me, I got new information about medical research, and which is very interesting to us. And finally, so please register. We will keep the registration link for 50 minutes after the end of the presentation. So if you'd like to get a certificate of attendance, you must register, and the certificate of attendance will be issued later in August due to time, busy schedule. And thanks for all. And thank you, Dr. Mohammed, and see you next week in the new seminar. Thank you, Dr. Mohammed. Bye-bye.
Assalamu alaikum.
Assalamu alaikum.